This is the Nutrients lecture with a focus on nitrogen for ENR4345, Methods in Aquatic Ecology course at The Ohio State University. In the first video in this module, you learned about the nitrogen cycle. In this lecture, you will learn forms of nitrogen commonly studied in freshwater, methods used to study nitrogen, the standard curve, as well as data interpretation for nitrogen samples. Nitrogen is essential for life because it is required for amino acids and nucleic acids. In many healthy aquatic ecosystems, nitrogen is a limiting or secondarily limiting nutrient. Limiting means that no more primary production can occur unless more nitrogen is added to the ecosystem. As you will remember from the previous video, bacteria facilitates the processes of nitrification and denitrification as part of the nitrogen cycle, meaning that bacteria is essential to maintaining the nitrogen cycle on Earth. Nitrogen fixation is the term for bacteria that are able to convert atmospheric nitrogen into ammonium, which can then be used by plants and algae. This photo shows a cyanobacteria colony, and the arrow is pointing to a heterocyst, which is a specialized cell where this cyanobacteria can undergo nitrogen fixation. Interestingly, lightning can also convert atmospheric nitrogen into nitrates when it strikes the ground. Similar to phosphorus, there has been an anthropogenic increase in bioavailable nitrogen in freshwater systems during modern times. This increase is due to artificial nitrogen fixation in order to increase crop yields. Some effects of excess nitrogen in a freshwater system include that it can facilitate harmful algal blooms and can be toxic both to humans and to aquatic organisms. As an example, nitrate can kill human babies when in high concentrations. This is called blue baby syndrome, and the infant cannot get enough oxygen. Another example is that high levels of ammonia, nitrate, or nitrite within an aquarium fish tank can harm the fish, and this is why you want to do regular nitrogen testing on your aquariums. There are different forms of nitrogen that we can measure in the water, and the type of nitrogen that we will measure will depend on our research question. The photo in this slide represents whole water that was collected with a grab sample. You may remember that we looked at phosphorus fractions from this same sample in the last module. As you can see, there are a lot of different forms of nitrogen that are possible to study in a freshwater sample. Ammonium is easy for aquatic primary producers to use because it can be directly incorporated into their amino acids without any chemical changes. Nitrate is also a commonly studied form of nitrogen. Primary producers will readily take up nitrate, however, it'll have to be reduced to ammonium within their cells before they can use it. Nitrate is also the form of nitrogen that we will be analyzing in the laboratory portion of this module. Next, I want to introduce a few different ways that nitrogen can be analyzed. The first is the ion chromatography method, abbreviated as IC. This method only works on ions, or charged particles, and is often cited as a good method for analysis of nitrate. In short, a material with the opposite charge to the ions that are being studied is loaded into the machine. The ions are attracted to this material as they pass through the machine and are removed from the sample when they stick to the charged material. Then, the machine reads a change in gradient, such as pH or specific conductivity, to quantify the ions removed from the sample. A colorimetric method can be used to analyze nitrogen. Similar to what you learned last week in the phosphorus module, the whole water sample can be digested, which renders all nitrogen within the sample reactive with the reagents used in the colorimetric method. For nitrogen, there are several different digestion methods that can be used to analyze whole water, including the Kjeldahl method, as well as alkaline persulfate digestion. After the sample is digested, there are a few different methods or different types of reagents that can be used to achieve results based on the portion of nitrogen that you are targeting in your sample. This process can be tricky and involves the use of strong acids. So for the purposes of this course, we are going to focus on analyzing the samples from the previous lab that we filtered with a 0.45 micrometer filter. Once we have filtered our sample, there are several different colorimetric methods that can be used, meaning we will use different reagents depending on the type of nitrogen that we want to analyze in our sample. The chemical reaction with specific reagents will create a color change reaction that is proportional to the amount of analyte in the sample, which then can be read on the spectrophotometer. For example, to analyze nitrite, we can add reagents for the Grice method to our sample, which will then react with the nitrate in the sample to create a pink or red color, proportional to the amount of nitrite in the sample. For this class, it is not important that you remember the names of each method used. It is more important to understand that different reagents are used for each type of nitrogen studied. Cadmium reduction is a commonly used method where the sample is run through a glass column containing cadmium, which will chemically convert all the nitrates into nitrites. This photo shows an example of a nitrogen sample that has an unusually high concentration, and it's being run on an autoanalyzer. The cadmium coil is this piece covered in red, and the sample will be run through it, and next it'll travel up the machine where the reagents for the Grice method are added, and time is given them for them to react and turn a pink color, which will then be read on the spectrophotometer, which is located just above the cutoff of this photo. The absorbance is read on the spectrophotometer to understand how much nitrite, both originally present and converted from nitrate, is in the sample. After analysis is complete, we can mathematically calculate the nitrate present in the sample by subtracting the nitrites from the nitrates and nitrites after it was run through the cadmium coil. Next, I want to talk about the 2,6-dimethylphenol method, which is the colorimetric method that is described in your laboratory handout and is the method that we will be using in the lab period. This colorimetric method is less commonly used because it cannot currently be automated with an autoanalyzer. This method is also more expensive than the other method because you're purchasing pre-made reagents in the kit rather than taking the time to make reagents yourself. However, some benefits of using this method are that it is less toxic, safer to use, and faster than the other colorimetric method. A more detailed protocol for this method is your required reading for this week. In short, you will be pipetting your nitrate sample into a vial containing reagents. The sample will turn a pink color, and the pink color will be proportional to the amount of nitrate in the sample. You will read the absorbance of the sample on the spectrophotometer, and then compare the absorbances of your sample to the absorbance of standards that we use to make a standard curve in order to calculate the nitrate concentration in each sample. In the last module, you learned about the importance of using a standard curve to interpret your phosphorus results. We will be using the same method to interpret nitrogen results. Please feel free to go back and review the last lectures if you need a refresher on this topic. I want to introduce the Beer-Lambert law, which is often shortened to just Beer's law. This states that the higher the concentration of a sample, the less light can pass through the sample. So this figure represents reading a nitrate sample on the spectrophotometer. You have a beam of light that's passing through the sample. Some of the light is absorbed by the sample itself, and some is able to pass through, where it's read by the detector on the spectrophotometer. The detector will then calculate the difference in light before and after it passes through the sample, and that will be the absorbance value. Beer's law tells us that this change in light is due to the concentration of the sample. Now that you have gotten your nitrogen results, it's time to think about data interpretation. Last week, we went over QA/QC protocols in the phosphorus lecture, which will be similar for nitrogen. Finally, I want to reiterate that your research question will drive the form or forms of nitrogen that you will want to study, as well as how many samples you will like to collect. For example, if you are interested in studying stream dynamics, it is probably important to study all types of nitrogen dissolved in the water over a longer period of time. However, if you are trying to protect babies from suffering from blue baby syndrome, one nitrate sample may be enough to determine if the water is currently safe to use.